Then why'd you ask it?
Because it didn't sound so stupid in my head.
Which brings me back to my question. Why'd we ask it? All right, let's move on. So anyway, Archaea and Protista. Let's start with Archaea. First of all, an interesting thing is the name. It shares a Latin root with a word we might use from time to time, actually. Does anyone know? I feel this about myself increasingly with each passing day. Old, archaic, right? Archaea, old. And that is so called because we know that they are kind of simple life. They resemble life as it sort of was in earlier time, right? Now, they still exist modern today. These are not some paleontological find. Like, they exist in the modern world, but they are a simple cell type, like bacteria. In fact, we say that they are prokaryotes. So they're morphologically similar to bacteria. In fact, for a long time, they used to be classified as a type of bacteria called Archaebacteria. But that's outdated. We no longer think that. The reason we no longer think that, Archaebacteria implied that we thought they sort of predated bacteria, but had survived through that time and evolved a bit. What we now know is that they're very distinct from them. But they are morphologically similar. Remind me what that term morphologically means? Morphos means shape, yeah. So, in other words, shape and structure. So in what ways is their shape and structure similar to that of bacteria? Well, the first thing is that they're prokaryotic. I'm not going to write everything that means, but I hope by now you have down what that means. What's it mean that they're prokaryotic? Yep. Simple, single-celled, good. Yep.
They have no nuclei or organelles.
Beautiful. Very good. Yep, exactly right. No organelles, no nuclei, simple, single-cell, unicellular, absolutely. They have similar motility structures, all right? What do we mean by that? What are the motility structures of bacteria again? Yeah.
The three ones are flagella.
Yeah, very good.
Pili.
Oh, look at you.
Cilia.
Very good. And I'm going to stick with flagella and cilia for now, because pili, I'm going to think of less of mobility as more sort of stopping motility, but absolutely. So they have similar motility structures. They reproduce similarly. Okay, so if all that is true, why are they different? Well, the first and most key difference is most of their cell walls lack peptidoglycan altogether. Do we remember that peptidoglycan or peptidoglycan that is found in the cell wall is found in all bacteria cell walls, though in roughly half of bacteria it's found on the outermost surface of the cell wall, and in others it's underneath another layer. Does anyone remember what test we performed that tells us whether they are one whose peptidoglycan is exposed or whether it's buried under another layer of stuff?
You like try and stain it?
Yeah, does anyone remember the name of the stain? That's exactly right. Because there's a lot of different stains you can do for a bacterium, but it is gram staining. Good. And if it takes gram staining, then we say it's gram positive, and that would mean its peptidoglycan is exposed. If it doesn't take the gram stain, we say it's gram negative, but that doesn't mean it doesn't have this peptidoglycan. It just means that that is under another layer of stuff, under a layer of these sort of liposaccharides. But for Archaea, they lack it completely. Now note, some still give a false positive for gram staining. So some still they will still take a gram stain, but it's actually a different chemical that's taking it. But mostly they're all gram negative. That is not a big meaningful difference, but it's noteworthy. The biggest meaningful difference is that DNA evidence, which is another way of saying that would be what type of evidence? Evidence. Sorry? Yeah, DNA or RNA, but what do we call that class of evidence when we're looking at that type of evidence to compare how related things are? Yeah, specifically, phylogenetic, where we're applying genetics.
Please excuse this
interruption. When it's convenient for you, could Kayla Douglas please come down to guidance?
Absolutely. Kayla, when it's convenient for you, you can head on down. This note will be online. We'll fill it in. So if you want to go now, go right ahead. Cool. So DNA evidence, which if you remember we call phylogenetics, which is using DNA evidence to classify how related things are, shows that they branched from modern bacteria just as early, and in fact, from modern bacteria, no later than the eukaryotes did. In fact, a little bit later. I should have said no earlier. No later is right. Yeah, that's right. That's what I meant to say. Sorry. In other words, despite the similarities, they're no closer related to bacteria than we are. They seem like it, they look like it, but they aren't actually more closely related to bacteria than we are, and that's why they're their own kingdom. Now, having said that, the Archaea took a very different evolutionary path than we did. We call this sympatric evolution, where despite the fact that they evolved from along their own path, they underwent a lot of the same evolutionary tendencies that bacteria did. Does that make sense? Even though they branched off very early, their evolution has been very similar. That happens sometimes in evolution, where two things can evolve along different paths but end up being quite similar to one another. You might use the example of bats and birds, right? Very different evolutionary paths, but actually quite similar, right? When you think about it. Yeah. Oh, of course I can. Is that better?
Yeah.
Great. So, despite this, they're similar in a lot of ways. And there's no point writing that because we've already talked about the similarities. All right, so what are they like? Well, the nice thing is we don't have to talk too much in detail because they're a lot like bacteria. Like we said, no peptidoglycan, so they're all, they're mostly gram-negative, and like we said, some false positives. They also undergo, what did we call the process by which prokaryotes divide asexually? Anyone remember? Grow bigger, grow bigger, grow bigger, divide in half? Yep. It is, but what do we call the specific mechanism? Anyone remember? Nope, mitosis is what we undergo. And mitosis, if you think about it, like involves all kinds of nucleus stuff and things like that. Yeah, very good. They're not going to have to undergo that, right? Because they don't have nuclei. They don't have all the same cell structures we do. So they also undergo binary fission. And if you remember those words, what does binary mean? Two. And what does fission mean? Break apart. If you think about like nuclear fission, which is like how an atomic bomb works and how an atomic reactor works, that's by taking atoms and breaking the nuclei apart, which releases energy. Okay, how do they move? Well, we see those same structures, right? So we're going to just say same structures as bacteria. But the main thing we're going to take away from these guys is that unlike bacteria, Archaea are often found in what we call extreme conditions. We say that many of them are extremophiles. Okay. Sorry about that. I just wanted to see if I had the list there somewhere. I thought I'd made this note differently. I wonder if I used an old copy. But that's okay, because we can handwrite. What are some extreme conditions on Earth in which we might find these fine fellas? Yeah. Freezing cold temperatures. Good. What else? So, and we find those in the Arctic and things like that. Where else? Hot conditions, right? Where else? What are some of the other extreme locations on Earth? So yeah, if you go like to the North Pole, the South Pole, you go to the Arctic, you get very cold conditions. Good. Dry conditions. Good. Absolutely. What about another one? Sorry? Yeah, that tends to be good for bacteria and cells. That is an extreme. That's kind of extreme for us. But by and large, bacteria and stuff like moist conditions, but even like extreme acidic conditions. So we'll give a few examples. We're not going to list every one. But for example, we get what are called thermophiles. What do we suppose where they typically thrive? They thrive in hot conditions. Good. Right? You should recognize thermo as a prefix that means heat. Right? It's where we get our thermos. It's where we get our thermostat. It's where we get thermometer. Right? Thermometer. Thermo, heat, meter, measure, heat measurer. If somebody was like, hey, pass me the heat measurer, you'd be like, that's stupid sounding. But if they said, pass me the thermometer, you'd be like, oh, they sound smart, but it's just a heat measure, if you know Latin. What else do we have? We have halophiles. Does anyone know what that means? Does anyone know what halo as a prefix means? Trick from the periodic table, we talked about the halides. I don't know if that helps or not. Anyone? Oh, sorry, did I hit you? I didn't mean to hit you with the chair if I did. Halophiles, does anyone know? They thrive in salty conditions. They thrive in conditions with extreme levels of salt. Yeah. Oh, so one would be like there's certain pockets of like oceans where there's extreme salt concentrations. And obviously lots of things can live in the salty oceans. Mind you, we can't live on salt water. Lots of things have made that adaptation. But where salty conditions get heavy enough, things just tend to die. Has anyone heard of the Dead Sea before? Literally so called because there's basically nothing living in it, though there's a bunch of Archaea living in it. It is so salty that A, things basically just can't thrive there, and B, it's famously so salty that like you don't just float in it the way you float in normal water. It's got such a salt concentration that like you lie down on it and you basically just sit on top of it like you're on a floaty, which is famously pretty cool. Because the reason you sink into water is that you're heavier than the water is. In other words, if you made yourself out of the same shape that you are but out of water, it would weigh a little bit less than you do. So when you sit there, you displace water. It's more energetically favorable for gravity to pull you down and push the water up. Why doesn't a boat sink? Because even though what the hull is made out of might be heavier than water, it's got a whole bunch of air inside, which is much lighter, right? So as long as it doesn't tip over and water can't replace the air, when it sits in the water, it weighs less than the amount of water it can displace. All the water a boat can push out of place is equal to its own weight. Does that make sense? So when you put a boat down in the water, it can move exactly as much water as it weighs. Does that make sense? So it's going to push, we call that its displacement. So it puts it down, it pushes down, gravity pulls it, and the amount of water it can push away is how much it weighs. If it pushes away enough water that it can get under the water, then you know what it's going to do? Sink to the bottom. It'll just keep pushing water out of the way, keep sinking. Keep pushing water out of the way, keep sinking. But if it doesn't, if it pushes water out of the way and is still above the water, then it just floats on top of it. That's what determines if something floats or not. Given its density and its shape, can it push enough water out of the way that the water can get on top of it and push it down? In that case, it'll sink. Or if it can't do that, it'll float. So we talk about the buoyancy of something as a function of its displacement. As it goes into the water, it has to push water out of the way, and it can only do that based on its mass. Did that answer your question? So when you add salt to the water, this is the second half of the answer, the water gets heavier. And as a result, you can't push as much of it out of the way. And if you can't push enough of it out of the way that it kind of laps up over top of you and get on top of you and push you down, then it can't get on top of you and push you down, and you float. So then you're like a boat. And haven't you always wanted to be a boat at least a little bit?
Yes.
A little bit?
Yes.
You know that Theodore the Tugboat? Have you guys ever seen this thing? Your parents and your little take you to see this thing. It comes through the canal once in a while. It's a big tugboat, but it's like painted like it's a person. It's being repaired, so it's in dry docks in St. Catharines. So every time I go down to the canal and take my dog for a walk, there's Theodore the Tugboat. That's the end of my story. It always makes me happy, but it makes me think I'd like to be a boat. I'd be a good boat. I'd be a happy boat.
Oh, that's fun.
Yeah. Sorry? Yeah, absolutely. And the saltier, the harder it is. Like, we like a little bit of salt. We need some, actually. Every living thing needs some salts. But when it reaches a certain level, it gets hard for cells to function. And when it reaches a certain function, it becomes basically impossible for cells to function. We have a lab we do in, I'm blanking for a moment. Is it grade 11 or grade 12? I did the membrane lab. Can't remember. Want to say grade 12. But anyway, we have a lab we do where we actually demonstrate that, where we actually like make cells out of like dialysis tubing, which is like semi-permeable tubing, and then put it in solutions of different salinity. At a certain point, if it's really salty on the outside, all the water just leaves the cell and the cell just shrivels and dies. And it takes specific adaptations to make a cell survive that. And we call something that can survive that a halo, meaning salty, phyle, meaning likes salt. This one I bet you can figure out. Acidophiles. Yeah. Yeah, they thrive in acidic conditions. Good. And then there's some things that require them to have all three traits. And I'm not going to sit here and list every possible combination. But for example, down like in the Marianas Trench and down in the deepest environs of the ocean, you have these places where there's subterranean geysers that produce extremely hot, extremely acidic, extremely salty water that just jets out. And like very few things can live there. But there's some archaea that thrive in exactly those conditions. So I'm not going to list them all. Just to give an example, you can have thermoacidophiles. Like, I'm not even going to write the definition. But like you can start to figure it out, right? Like this thrives in hot acidic conditions and so on and so forth. Yeah. Yep, some do. Again, I'm not going to list every combination, but you get the idea. For whatever reason, bacteria have mostly outperformed archaea. There's a lot more bacteria on Earth than there are archaea. And I couldn't tell you why that is. It just happened. That's how evolution went. Just like humans thrived and Neanderthals didn't, right? I don't really know why humans thrived and Neanderthals didn't. Presumably we were better at killing them than they were at killing us. I don't know. But we did. Bacteria mostly get by in the world better. But there are certain places where bacteria do very poorly. Very hot conditions, very cold conditions, extremely acidic conditions, extremely salty conditions, where there are species of archaea that perform very well. And in those places, they have found their biological niche. Do you remember that word from grade 9 science? Does it help that I'm making a little hand motion like this? They found their niche. What's that word mean? Yeah. Sorry? Absolutely, a hobby is an example of this. A niche is literally finding your... Usually, though not always. Sometimes it can be more general. Some things, the niche is really broad. For most people, it's specific. Yeah, their kind of role, their place in the world. Does that make sense? And for most species, they have a specific niche. They have sort of a specific set of conditions that are both the environment, but also what animals are there and all that stuff. They have this place in the world that they fit where they are stable, where like how much they reproduce is balanced by how much they are killed and how much they die. How much they consume is balanced by how much the ecosystem can produce. And when a species finds a stable niche, they find a place where like they thrive not just for a little bit, but for a long time, because they exist there. Not only do they exist in a place that's well suited to them, but they exist in a place that's well suited to them and can sustain them over a long term. That's what we consider an ecological niche. Anyway, Archaea, largely their ecological niche is these extreme conditions. That tends to be where they survive and thrive best. Next, we'll talk about protists. And I have a textbook section here that we'll read after. I gave you both these, right? Or they gave you both these? Boy, I'm just noticing these did not print nicely. I am looking forward to actually getting textbooks. Can you all read that? Boy, I'm going to need my old reading glasses. That's what I'm going to need.
Some paragraphs are easier to read than others, which is annoying.
Yeah, that is annoying.
Yeah, the little tweets.
Yeah, it looks like crap, frankly. It's really frustrating.
Yeah, it's like the pictures are awful. Like they were written on the window and just went.
Yeah. Anyway, it's all good. By the way, I did want to mention, if you do look at the textbook section on Archaea, which you'll find in the first section on that has this on the cover, these kind of questions here, the prokaryotes, eubacteria, and archaebacteria, Archaea. That's on the bottom of page 51, and they literally just have a couple paragraphs about Archaea, and they move on. They mentioned a couple other types here. Methanogens. These live in low oxygen environments, and they can generate energy by a totally different method where they convert chemicals in the environment into methane gas, and they produce energy that way. So a totally different way than we make energy, than we get ours from plants, right?
Couldn't that mean they just survive in space?
Yeah, potentially, given, other things. These also have psychrophiles, which we didn't mention, would love the cold. But anyway, the section on Archaea is very short. But I want to talk protists. I want to talk Protista. So really quickly, bacteria, where are bacteria found?
Everywhere.
Yeah, they're found everywhere. What types of cells are they? Prokaryotic. This is just a little bit of review I built into this lesson. Are they single or multi-celled? Single-celled. Do they have organelles? No. How do you know they don't have organelles? Because they are prokaryotic, so they can't be multicellular, and they can't have organelles. And do they have a nucleus? No. Okay, there we go. That's just review. But protists, on the other hand, what type of cell are protists? We'll come back to environment. They're eukaryotic. Good. And that means that they are single-celled, or they can be multicellular. All right, so protists are the first thing we're coming across. Because they're eukaryotic, some examples of these are actually multicellular. All right, do they have organelles? Yeah. No, all. They all do. They all have organelles because by definition they are, even if they're single-celled. Yep. And do they have nuclei? Yes. And remember, organelles aren't organs, they're organelles. They're the units inside the cell, right? The mitochondrion, the chloroplast, the this or that. Now, what's their environment? To be clear, they can be found all over the place, but they tend to be one universal truth, is they're always found in moist environments. In moist or aquatic environments. What does aquatic mean? actually in water. And obviously moist means like water present, right? Like damp, things like that. What do they eat? So some are autotrophic. Remind me what that word means. Sorry? Autotrophic, yeah, produces their own food, which generally means undergo what process? Photosynthesis, right? So some are autotrophic, they undergo photosynthesis, but most have to consume other organisms for nutrients. In other words, we say that they are what? Hetero, not zygous. Heterotrophic. Yeah, good. Very good. This comes in a few forms, but many are what we call parasites. What's a parasite? Or parasitic? What's it mean for something to be parasitic? Yeah. They feed off a host. Exactly. So it's different. It's not like us, we kill an animal and then we eat it, right? If you're a parasite, you don't kill the animal, you just kind of move in. You know, imagine if you just were like, hey, instead of like, I don't know, like killing a cow and eating it, what if I just like, you know, made myself a bedroom on its back and just chowed down a little every day? That's what parasites do. It would be very impolite behavior if you're human. But for small little single-celled organisms, it's pretty common. So many are parasitic, but by no means all. Yeah. There's a great variety of food sources outside of that. We don't have time to go through every type. Your textbook has a slightly larger list of types. So you can see page 63 from that. Page 63 has a bigger list of types, but I want to focus on key types that you might come across. And so the first one that comes along is algae. Nope. This is an example of not being parasitic. Algae are protists, except for blue-green algae, except for the specific one called blue-green algae, which is not. They can be single-celled, but mostly they're multicellular. And they're autotrophic, meaning they photosynthesize. Where do we tend to find algae? Yeah, in water, right? And specifically, they tend to do well in places with somewhat stagnant water because they don't have root systems to the same degree that plants do. They don't latch on as well. They don't do as well in like moving water as plants do even, though even plants don't do well in fast moving water. So sort of the more stagnant a water source is, the better algae do. This is why, like, you know, they have you add like the pump, right, to your, if you have a fish tank or if you have a pond or if you have a pool, you want to keep that water moving to prevent algae growth. Does that make sense? Well, again, for that reason, yeah, because algae will grow, but so will other protists, so will bacteria, so will other things, and you just get things that are toxic or parasitic even. Yeah. Like if it dries out, will it die? Yeah, absolutely. Yeah, and if you've ever, as I remember, that my grandparents used to have a pond, and we would skim the algae from it a few times a year if it started to build up. And if you skim it out and just leave it on the lawn, it just kind of turns brown and dies up. Yep, absolutely. Okay, another example. These lovely fella, slime molds. They're pretty gross, right? So at one point, they have a unicellular stage and a multicellular stage. And so when they first grow, I shouldn't say is still, but can still be. Like this example here is still unicellular, even though it's quite big. So it has a unicellular stage, and then eventually this will actually split into individual cells, and they have a multicellular stage as well. They're large. Aren't they lovely looking things? Right? All right, and then this one you've probably heard of before. What's this? Yeah. No, I was gonna amoeba, or amoebae or amoebas, which we tend to actually just now pluralize with the S as if it were an English word, are really cool things. What you see here is drawings not of different amoebae, but of the same amoeba, observed over time. And this was taken with an early microscope in 1755, so they drew what they saw. And what do we see about this thing while it's doing what it's doing? Its cell is just dramatically changing shape. So these are really neat because these move not with flagella or cilia. And by the way, this is not true of every protist. This is true of amoeba. All right, there are plenty of protists that do have cilia. There are plenty that do have flagella. But these move not with cilia or flagella, like some protists, but with what we call pseudopodia or pseudopods. What's a pseudopod? Just imagine this. Imagine you're just a big blob, and if you need like a leg to walk that way, you just like, whoop, here's a leg, and then you pull yourself that way, and then like, whoa, here's a. They just grow and extend their cytoplasm in every which direction, and so they can use it to kind of swim, but they can use it to pull themselves up and over hard surfaces or things like that. They can just change their shape willy-nilly. So this is a single cell, and you can see it just basically changes shape as needed to move. What does pseudo mean? We often use it in the guise of that guy's, we talk about pseudoscience, things being pseudoscientific or pseudo, yeah. Sorry? Imitation, good, fake, not real. What about pod? What's a podiatrist? Pod. Yeah, feet, legs, limbs, right? So pseudopods is like fake legs. It makes little fake legs. Again, things sound scientific when they're in Latin, right? And then you translate it and you're like, oh, wait, how does this move? Fake legs. Sorry? Extensions of the membrane, yeah. Sorry, you can't tell that because that's the worst handwriting that any human being has ever done. But that's a little better. These are heterotrophic, and they tend to feed by simply engulfing their food. In a process called phagocytosis. And protists are almost a category of just exclusion. Sometimes we talk about that, where we talk about some diseases are diagnoses of exclusion. Have you ever heard that expression before? In other words, there's some diseases where we basically say, okay, this person's come with these symptoms, and we try all these other things we know cause those symptoms, and finally, if we can't figure out why you're this, anyone ever heard of chronic fatigue syndrome? We don't know what causes it. But basically, if somebody comes and they're tired all the time, and like exhaustively, and we check all the things we'd normally check for, we check their blood level, their blood iron levels, it's good. We check this, we check that, and we check everything, and we kind of can't figure out why they're like that. We say, okay, you have chronic fatigue syndrome. Literally, you're tired all the time, and we don't know why. Does that make sense? Protists are this thing where we say, like, what's a protist like? They're all so different from each other. Basically, a protist it's a eukaryote, but it's not a plant, it's not an animal, and it's not a fungi. If it's a eukaryote, and it's not a plant, an animal, or a fungi, we say, yeah, that is a protist. It's just kind of this really broad category, which makes it hard to talk about them broadly. See how different these look? It's everything from little single-celled things that change shapes and move like that and engulf things to slime molds to algae. Like, it's all over the place. But if you are a eukaryote and you're not a plant, an animal, or a fungi, then we say, yeah, it's a protist. It's not quite that simple. They also have to be genetically related to the other protists, but it's kind of the broadest group. It's the group that has the least in common with itself. Does that make sense? Fungi would be number 2 for that, but protists definitely are number 1 in terms of there's not a lot of common characteristics between protists. They've evolved to be very, very, very diverse. How do they reproduce? Well, because they are eukaryotic, they undergo mitosis, which you learned in grade 10 science, and we will learn again when we do our genetics unit. They undergo mitosis. Really quickly remind me of the phases of mitosis. Interphase, so that's before mitosis, but yeah. Which one's first? Prophase. Metaphase. Anaphase. Telophase, telophase, yeah, and then cytokinesis, then back to interphase. Good, very good. They undergo mitosis. Yeah, that's the main thing to know. So mostly asexually, right? But just like bacteria, there are variations. I'm not going to go through it. Budding is different than binary fission and mitosis. In mitosis and binary fission, the cell kind of grows and it basically splits in half, right? You get 2 cells that are of equal size. There's a variation on this called budding, where the DNA will replicate. It will move to kind of a quarter of the cell, and a very small section of cell will break off. And then it will grow, which is almost more like how we produce, right? Where we produce a small little thing, little babies, and they grow into people over time, right? So in budding, the new DNA would get put in a little tiny section of membrane and released, and then it would grow to its own full size. That's a variation that we see sometimes. They have their own forms of sexual reproduction, which are similar to bacteria, and that involves two cells joining up and exchanging genetic information. We're not going to get into specifics here, in part because there's just so much variation within Protista. Is that fair? So we're not going into specifics. And we already mentioned flagella, cilia, and pseudopodia. I'm not going to make you draw that now or go into it. That's the basic there. We don't need to know a ton about these things. I'd rather you learn about them by learning about an individual species, generally speaking, which you're going to do in what project? The project I gave you yesterday. For those of you who are here, what is that? Yeah, go. Well, they're all diseases. You're going to study 3 diseases. One that is bacterial in nature. It's caused by a bacterium, absolutely. One that is viral, and the final one that is caused by a protist, right? Very good. So I like this as a better way of kind of focusing on this, because again, there's just so much variety there. The details for that can be found on Teams. It is not due until the end of the unit. Tentatively, that is not the coming Monday, but the Monday after that. But if we need a bit more time, we'll take it. If we happen to end up a day or two ahead of that, we'll end up a day or two ahead of it. I would never move that deadline back on you. I wouldn't be like, now it's due Friday. I won't do that. But I might do, now you have until Wednesday. In other words, if I set a deadline, I may end up giving you more time. I won't give you less. I will never do that. That wouldn't be fair, right? That would obviously be unfair to be like, haha, you thought you had till Monday. You don't. It's due yesterday. You're all late. See what I mean? You would do that. This is why we're enemies. So anyway, you can work on that with time that remains. And I know we don't have the computer's book, so if you wish to use your phone for productive purposes, you may do so. If you use them for unproductive purposes, who will your phone get to meet? Barry. It'll meet Barry. So you have that. You can start your disease project. You also have something that is due next week, due next Wednesday, which is your dichotomous key task. Good. That's okay. So you have lots to keep you busy. And then the other thing I would say is, oh God, there's so many hands. Just let me finish my sentence and I'm all yours. The other thing I would say is the 2 textbook sections I gave you today, they go into a bit more. Sorry, just let me know when it's good for you. Is it good for you? Can I talk now? Oh, good. I appreciate that. The textbook sections I gave you, I want you to, over the course of the weekend and Monday, Tuesday, make sure you've read these. All right. And again, am I giving you any specific questions on them right now? No. I'm going to tell you right now, there's a little bit of information there, and I'm not talking hyper-specific stuff. All right. But there's a bit of general information in there that is fair game, and I'm intentionally going to kind of build in to our next quiz. And then we'll talk about it's nothing major. And it's not going to make the difference between like, oh, I would have had a 99, but now I've got a 7 in the course because I didn't read that. But it'll be little points of detail that'll make the difference between, say, an 85 and a 95 kind of thing. Does that make sense? And it's not that you'll have had to sit down and memorize this in detail. If you give it one good reading, but make it an active reading. What's my advice for how to go about reading? Use some highlighting, take some notes. Another good one I haven't mentioned yet, but this is my wife's questions. She's a big believer that a good way to actively read is as you read something, have a notebook there, and anything that you think either, ooh, I wonder about this, or, ooh, I didn't know that, but don't write it here as a fact. Don't write it here as like, oh, I didn't know that amoebae moved by like pseudopods. She wouldn't write, amoebae can extend their membrane in cytoplasms to form pseudopods. Rather, she would write, what are pseudopods and how do they make amoeba move? Do you know what I mean? She just writes, A, that's quicker because it doesn't take as long to write, so you can keep reading faster. But B, when you reread your notes, it doesn't have the answer in it, interestingly. It only has the question, which actually trains your brain not to keep seeing that information, but instead it trains your brain to see a question and go, oh, I remember that. I read that. It's pseudopods are the little bits that stick out of the protist and help it move. Does that make sense? So by making a sheet that doesn't have a list of things you learned, but rather has a list of questions that remind you of the thing you learned, you train yourself to remember what you learned. That's a good trick. If I'm being honest with you, it's not one I've learned. It's not one that I've used, but I think it's a really good one, and my wife's a lot smarter than I am, so like I trust her. I think like if she says I do it this way and I say I do it this way, I'd go with her. She's the brains of the operation. So you can work on that. You can do your readings. You can work on your yeah, that's the one. Or you can work on the disease thing project. And again, you can read the details about that on Teams. I'll leave it to you. Yeah.
Did you ever go and get those Bristol boards for them?
Yes, I did. Now you might ask a follow-up question. Oh, can I have it? And then I would say, no, because where are they? And you can't make this up.
Oh, you're lying to me.
They're on my desk in my office, which I call it my office. Really, it's my laundry room where I built a countertop that I use as my desk. During COVID, I called it the Sadness Cave, and I recorded all of my lessons from the Sadness Cave. And now even stepping into that room makes me upset. But anyway, none of which has anything to do with the fact that I have a big old Bristol board sitting there. So I would say plan around it, and I will make sure to have it to you on Monday. And if I don't have it to you on Monday, I will give you a 20% bonus on this project.
Oh,
there you go.
okay,
So now I got to get it to you. Now I got to get it to you. Right now I don't even have a choice because I don't like to be nice. But I mean that. You can hold me to that. But you'll also have to remind me because I've already forgotten that I said that. Yeah. 110%. Yeah, nothing I can do about that. Yeah. Okay.